The goal of this experiment is to resurrect dormant stages of Daphnia Magna species. This is a powerful method that dramatically advances data collection to generate long-term data in natural systems.
This method enables the study of ecoevolutionary processes which occur over extended periods of time in nature within the time frame of a research project. The main advantage of this technique is that it provides unprecedented power in dissecting the processes and mechanisms of evolution if applied to populations that have experienced changes in selection pressure over time.
Although the methodology of resurrection zooplankton species have been frequently applied, so far it has been propagated among laboratories only via directed knowledge transfer. Visual demonstration of this method will allow non-experts to apply resurrection ecology by following a step-by-step guide, promoting the application of a powerful technology to facilitate the study of evolution.
After obtaining a core tube of sample sediment from a lake or pond, use a flat metal tool to slice the core into horizontal 1 cm layers. Then, collect the sediment layers in separate bags.
The Big Bang corer is designed to reduce disturbance of the sediment layers during sampling. When other corers are used, the outer ring of each sediment layer might be removed with a cookie cutter sort of blade to limit contamination among layers.
Finally, store the sediment samples in a sealed cardboard box in a dark room at 4 degrees Celsius.